Hello, everyone, and welcome to today's live broadcast, Characterization of Immune Checkpoint Expression and Infiltrating Immune Cells in Tumor Microenvironment by RNAscope ISH. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by labroots.com, the leading social media site for science professionals, and sponsored by Advanced Cell Diagnostics, a biotechnology brand. Advanced Cell Diagnostics is a world leader in RNA biomarker analysis for precision medicine, providing the most advanced RNA analysis solutions for cell and tissue-based research, drug discovery and development, and diagnostics in molecular pathology. Its proprietary award-winning RNAscope technology is capable of detecting and quantifying RNA biomarkers with single molecule sensitivity and single cell resolution in routine clinical specimens. The technology has been employed in more than 1,000 peer-reviewed publications by a rapidly growing customer base of thousands of active users. The company builds strategic alliances with its customers and partners to develop companion diagnostic tests for existing and emerging therapeutics, and also pursues internal programs to develop and commercialize proprietary cancer diagnostic tests. To learn more, visit www.acdbio.com. Before we start, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. And if you can't hear or see this presentation properly, let us know by clicking on the support button at the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenter. We are proud to welcome Annelies Laeremans, Ph.D., a scientist in R&D at Advanced Cell Diagnostics. The Applications Group manages numerous projects within the R&D laboratories and with collaborators from around the world. They demonstrate new and exciting applications of the RNAscope and BaseScope technologies, and also their ability to detect RNA biomarkers with single molecule sensitivity and single cell resolution within the morphological context. Prior to Advanced Cell Diagnostics, Dr. Laeremans completed postdoctoral studies in cancer biology at the University of California, San Francisco. She received a Ph.D. in cognitive and molecular neurosciences from the University of Leuven in Belgium after completing a one-year visiting scientist position focusing on medical entomology at the University of Florida. Previously, she graduated as a bioengineer in cell and gene biotechnology from the University of Leuven with a 6-month international exchange program at the University of Milan in Italy. I will now turn the presentation over to Dr. Laeremans.
Thanks for the introduction and welcome to the webinar, everybody. Today I will discuss the characterization of immune checkpoint marker expression and infiltrating lymphoid and myeloid immune cells in the tumor microenvironment, and how the RNAscope in situ hybridization technology can aid in characterizing the TME. Here's an overview of today's webinar and the different topics we will cover. Before showing the results of our immuno-oncology proof-of-concept study in non-small cell lung cancer and ovarian cancer, I will give you a description of our RNAscope ISH technology that was used to generate the data discussed throughout this webinar. Our proof-of-concept study is built around two main topics: the expression and co-expression profiles of therapeutic checkpoint targets and the immune infiltration and state of lymphoid and myeloid cells in the TME. Regarding the immune checkpoint markers, we will show data on the visualization and quantification of checkpoint expression within the morphological context of the TME, on cell type-specific expression of PD-L1, on heterogeneous expression patterns of some immune checkpoint markers, and finally on immune checkpoint co-expression at the single cell level. We will move on to the RNAscope ISH technology. Here you can see a brief overview of the technology. It is a slide-based assay intended for the same types of samples that you would use for IHC or immunofluorescence. This can be either FFPE, so formalin fixed, paraffin embedded, or fresh frozen or fixed frozen tissue sections or cells. The RNAscope methodology involves hybridizing the target-specific oligonucleotide probes to the target RNA. And the target probe is a pool of oligos that is designed using established informatics tools to provide an assay which we guarantee will be single target specific. Detection is enabled through an amplification procedure that involves sequential hybridization steps to build up a tree-like structure shown here schematically, and I will describe it further on the following slides. Through this method, we are able to localize a tremendous amount of signal to an individual transcript to the point where we can actually detect and visualize individual transcripts as dots. These punctate dots allow for the subsequent quantification of the detected gene expression. The first key feature of the RNAscope technology is the probe design. We depict the oligonucleotide target-specific probe as these to emphasize the fact that they have two regions linked by a spacer. The bottom of the Z complements and hybridizes to the target transcript, and the top of the Z is the base for the amplification structure. When two Zs hybridize in tandem to the target sequence, it creates a unique binding site upon which a pre-amplifier can bind and the amplification tree can build. A standard RNAscope probe for a target sequence of 1,000 bases or more will consist of 20 ZZ pairs pulled together and designed to hybridize next to each other along a target region. This allows for a tremendous amount of amplification and signal potential. However, only a few Zs are needed to bind to the target RNA sequence in order to generate enough signal for molecular detection. The second key feature of the RNAscope technology is the actual signal amplification. As mentioned, after hybridization with the target RNA, each Z pair creates a binding site at the upper portion of the Z for the signal preamplifier to bind. Each preamplifier can bind multiple amplifiers, and each amplifier can further bind multiple labeled probes. Labeled probes can contain a chromogenic enzyme such as HRP that generates a visible signal after chromogenic reaction with a substrate such as a DAB, and then it can be visualized under bright field microscope, or labeled probes can also contain fluorophores to visualize the signal under a fluorescent microscope. Lastly, background is suppressed by the RNAscope assay because the signal is dependent on two Zs binding next to each other on the target sequence. If both Zs do not bind, then the preamplifier cannot form a stable hybridization at the top of the Zs, and the amplification tree cannot be built. Consequently, no amplification of non-specific hybridization occurs, resulting in little to no background signal. Combined, the unique probe design and the amplification system ensure a high signal-to-noise ratio. The growth and adoption of the RNAscope is best exemplified by the number of peer-reviewed publications. We had our first publication in 2011, and since then there are over 1,000 peer-reviewed publications using RNAscope in high-tier journals. The graph to the right shows how RNAscope is highly relevant across multiple fields of research, with over 40% of publications in cancer research, followed by neuroscience and infectious diseases. This slide presents an overview of all the RNAscope assays that are available from ACD. We have the singleplex chromogenic assay for a single target available for both brown and red in manual and automated Ventana and Leica platforms. RNAscope is also available as a chromogenic duplex assay to visualize the gene expression of two targets simultaneously. And to visualize 3 or more targets, the RNAscope multiplex fluorescent assays V1 and V2 can be used. Images for these assays will be shown throughout this webinar. Here we show the typical configuration of an RNAscope experiment. So provided that the target is expressed in the sample and the protocol is followed as recommended, the performance of the RNAscope assay is guaranteed. Because tissue samples can be highly variable, proper sample preparation and the right controls are essential to an RNAscope experiment. Tissue samples must be properly fixed and prepared so that there is good RNA quality in the sample and no background due to poor fixation. ACD provides guidelines on how tissues should be fixed for optimal performance on our assay. Here you can see 3 serial sections from a human lung cancer sample on which we performed the manual red chromogenic assay. On the left is a section probed for our negative control probe DEB-B, which is a bacterial gene that is not expressed in animal tissues. As observed in this sample, there should be no signal for this probe. In the middle panel is a serial section from the same sample stained with our positive control probe for the housekeeping gene PPIB. As observed here, you want to see fairly uniform target detection with the positive control probe, indicating good RNA quality. From the results for these control probes, we can conclude that there is good RNA quality throughout the tissue sample and that there is little to no background, so we can be confident that the localized pattern for the target probe, PDL1, shown on the right, will in fact be correct. Next, we will cover our immuno-oncology proof of concept study with data for both non-small cell lung cancer and ovarian cancer. The generation of immunity to cancer is a cyclic process that leads to the accumulation of immune stimulatory factors that should amplify and broaden T-cell responses. The cancer immunity cycle is also characterized by inhibitory factors that lead to immune regulatory feedback mechanisms which can halt the development or limit the immunity. Although infiltrating T cells have been correlated with improved clinical outcome, they are ineffective in eradicating tumors due to their inhibition by immune checkpoint molecules. In addition, immunosuppressive factors in the local tumor microenvironment contribute to tumor evasion by recruiting and modulating regulatory T cells and myeloid-derived cells. Immune checkpoint inhibitors have demonstrated therapeutic efficacy and durable response for several tumor types, including non-small cell lung cancer. However, the majority of patients are either not responsive or relapse after initial response. Characterizing the TME for checkpoint expression with single cell and spatial resolution may provide critical insight into new immunotherapeutic strategies and identify new predictive biomarkers for stratifying patients most likely to benefit from these immunotherapies. Here is a schematic of the design of our proof of concept study. So in blue, you can see the processes preceding the RNAscope assays. First, FFPE tissue blocks are generated and undergo quality control by testing our negative and positive control probe as discussed in the RNAscope technology section. After screening for RNA quality, tissue microarrays are created for both non-small cell lung cancer and ovarian cancer. In orange, you can see the different RNAscope assays that were run. Besides single-plex data for PDL1 amongst other targets, we ran several checkpoint marker probe pairs, including PDL1, combined with TIM3, LAG3, CTLA4, and PDL2. The multiplex ISH IHC assay was used to visualize 3 checkpoint marker targets, being PD1, PDL1, and TIM3, simultaneously with the CD45 antibody as a marker for immune cells. The chromogenic duplex assay was also run for probe pairs indicative of different lymphoid and myeloid cell types and states, as was the multiplex ISH IHC assay for interferon gamma, CD4, and FOXP3 mRNAs combined with the CD8 antibody. In this first data section, we will highlight the expression and co-expression profiles of therapeutic checkpoint targets within the morphological context of the tumor microenvironment. Here we illustrate the RNAscope assay applied to non-small cell lung cancer tissues for checkpoint marker evaluation. The lung cancer TMA has 60 patient course, with 56 representing tumor tissues and 4 normal tissues. Duplex data was generated for these checkpoint marker probe pairs on serial sections. In the lower panel, the expression analysis is illustrated for 1 core. On the left, you can see the core probed for 2 checkpoint targets in red and green, with a higher magnification view of the selected area. For the selected area, you can discern how the Halo software identifies and segments individual cells and quantifies the number of dots for each of the 2 probes. Cells exclusively expressing one of the probes are shaded in green or red, and cells co-expressing the 2 targets are highlighted in yellow. We analyze the expression data as the % positive cells expressing selected checkpoint molecules. The result of this quantitative analysis is shown here as a heat map of selected cores with red and green indicating high and low expression levels respectively. PDL1 expression across the cores is shown in the top row, and % PDL1 positive cells was used to sort the cores. In the next slide, we will focus on these 4 selected cores to visualize the dynamic range of PDL1 expression. Here we visualize the tumor expression of PDL1 across 4 selected cores to exemplify the dynamic range of expression shown in the numerical table on the previous slide. Core 2B2 clearly demonstrates high PDL1 expression, with core 2C4 on the other end of the spectrum with very low to no PDL1 expression. In between cores 1G3 and 2G3 show medium-high and medium-low PD-L1 expression in the tumor respectively. Tissue core 2E2 is a hot or highly inflamed tissue core with lots of immune cells infiltrated in the tumor microenvironment, while core 2F1 is cold and shows a low level of immune cell infiltration. For both cores, representative images for the quantitative analysis of each checkpoint marker displayed by the rows will be shown in the upcoming slides. This image from core 2E2 visualizes the quantified checkpoint expression data for PD-1 in red and PD-L1 in green within the morphological tissue context of the TME. As indicated by the red shading in the table, PD-L1 and PD-1 expression are high in this highly inflamed core. While red PD-1 dot signals are present in the stroma, presumably on infiltrating immune cells like tumor-infiltrating lymphocytes, green punctate dots for PD-L1 mRNA expression are visible in tumor cells in addition to immune stromal cells. With this being a higher magnification image of the selected area, where stromal and tumor areas are indicated by S and T, and you can see the higher magnification over here. Cell type specific checkpoint marker expression in tumor versus stroma is very valuable information that, while not deductible from a heat map as shown, is clear from the stained tissue side, what you can see over here. In this cold tissue core with very low inflammatory cells infiltrating multiple tumor and stromal areas are visible, with one of each highlighted. In contrast with the hot 2E2 core, expression for the checkpoint markers PD-1 in red and PD-L1 in green is low to no, which can be seen in this representative image over here. This image from core 2E2 visualizes the quantified checkpoint expression data for PD-L2 in red and PD-L1 in green within the morphological tissue context of the TME. As indicated by the red shading in the table, PD-L2, like PD-L1 expression, is high in this highly inflamed core. Green punctate dots for PD-L1 mRNA expression and red dots for PD-L2 expression are visible in both tumor and stromal areas, which is especially clear from these higher magnification insets. Also noticeable is the co-expression of PD-L1 and PD-L2 in the same tumor cells, with arrows in both insets indicating some tumor cells with PD-L1, PD-L2 co-expression. Combination of the same checkpoint markers in this cold tissue core with multiple tumor and stromal areas indicated shows low to no expression for these targets. Moving on to the next pair of checkpoint markers, for core 2E2, LEG3 dot signals are shown in red, again combined with PD-L1 mRNA expression in green. While PD-L1 is expressed in both tumor and stromal cells, LEG3 expression is most prevalent in immune stromal cells. LEG3 and PD-L1 appear to be co-expressed in immune cells, with some indicated by the arrows over here. Like previous checkpoint targets, also LEG3 mRNA expression, again combined with PD-L1, is very low in core 2F1, characterized by a low level of immune cell infiltration. Here we show PD-L1 expression in green combined with TIM3 mRNA expression in red for core 2E2, so the highly inflamed core, while PD-L1 is expressed in both tumor and stromal cells as shown in previous probe combinations. TIM3 expression is most prevalent in immune stromal cells. What also can be appreciated from this image is the visualization of the immune infiltration into the tumor area, with TIM3 and PD-L1 co-expression in some immune cells, of which we also highlighted some with the arrows over here. When we move on to the cold core 2F1 for these targets, we see that similar to previous checkpoint marker expression patterns, expression for TIM3 mRNA in this low inflamed core is minimal, with some TIM3 positive cells shown in this higher magnification inset. In this last probe combination with PD-L1 for core 2E2, we show CTLA-4 checkpoint expression in red. While PD-L1 is expressed in both tumor and stromal cells, as shown in previous combinations, abundant CTLA-4 expression can be observed in immune stromal cells, which is highlighted by this higher magnification image. When we look at the same targets for the cold core 2F1, we see that there's a very limited number of CTLA-4 or PD-L1 positive cells, as is also shown by the shading in the numerical table. Next, we will briefly highlight the cell type specific expression in tumor versus stromal cells for checkpoint marker PD-L1. As exemplified in the previous section, PD-L1 mRNA expression was demonstrated in tumor cells and some immune stromal cells. In this duplex RNAscope assay, PD-L1 expression in green was combined with the tumor antigen c-Met in red. Co-expression of PD-L1 with c-Met indicated by, but not limited to, the arrows, confirms PD-L1 expression in tumor cells. Note that here, and also on subsequent slides throughout the webinar, arrows are pointing out some examples of cells co-expressing both targets, but they are not limited to those cells pointed out. Here we show two c-Met positive tumor cores with c-Met in red, and core 2E2 on the left is a PD-L1 positive tumor, while core 2A2 on the right is a PD-L1 negative tumor with no co-expression of PD-L1 and c-Met. Moving on to the next duplex assay, PD-L1 expression in green was combined with the tumor cell marker cytokeratin 19 in red. Co-expression of PD-L1 with cytokeratin 19 confirms PD-L1 expression in tumor cells, which can be seen over here. Here we show two keratin 19 positive tumor cores in red, and core 1B1 on the left is a PD-L1 positive tumor with co-expression of PD-L1 and cytokeratin 19, while core 1E4 on the right is a PD-L1 negative tumor with no co-expression with the keratin 19. In contrast with the two previous tumor cell markers, this duplex RNAscope assay combines PD-L1 expression in green with the immune cell marker CD45 in red. While PD-L1 is predominantly expressed in tumor cells, co-expression of PD-L1 with CD45 confirms PD-L1 expression in some immune cells, for which we highlighted some with these arrows. Also shown here is a PD-L1 positive tumor for two cores on the left-hand side of the slide, and you can see PD-L1 negative tumor for 2C3 and also core 2B2 on the right-hand side. Another feature besides cell type specific expression that is not visible from the heat map is when immune checkpoint markers have a heterogeneous expression pattern. In this section of the webinar, we will show some examples for heterogeneous PD-L1 and PD-L2 expression. In this image, PD-L1 mRNA expression is shown in green, and the two delineated tumor areas display different levels of PD-L1 expression, pointing to a heterogeneous expression pattern for this target. Also note that RNAscope shows no signal when RNA is lost in necrotic tissue with no intact cellularity, like visible on the right-hand side of this image. A similar heterogeneous expression pattern for PD-L1 in green can be seen in this specific core, with again areas with different levels of PD-L1 expression highlighted by the boxes, and also in this next example, where there's clearly different levels of PD-L1 in these two tumor foci. A duplex assay for PD-L1 in green and PD-L2 in red for the highly inflamed core 2E2 also demonstrates heterogeneous expression throughout the selected area for both markers, as illustrated by the 4 higher magnification images on the bottom. A different area higher up in the same tissue core also is characterized by heterogeneous expression for both immune checkpoint markers. Again, higher magnification insets on the bottom show this heterogeneous expression pattern for these targets. In this last section of immune checkpoint marker expression profiles, we will further elaborate on the co-expression of checkpoint markers at single-cell resolution. For this part, I will focus on PD-1, TIM3, and LAG3, visualized by the chromogenic and multiplex fluorescent IHC assays. This chromogenic example illustrates the single-cell co-expression of TIM3 and PD1 in red and green respectively, with some cells pointed out by the arrows. This type of expression analysis could potentially offer insights into why patients become resistant to single-molecule checkpoint blockades on one hand, and also on combinatorial therapies simultaneously targeting multiple immune checkpoint markers on the other hand. A similar co-expression pattern can be seen across multiple cures. So here we show you another example and also a higher magnification zoom of this example. And again, the arrows point to cells co-expressing PD1 and TIM3, but you can also see that there's multiple other cells co-expressing these two targets. Immune checkpoint co-expression patterns were also visualized by multiplex fluorescent ISH for 3 RNA targets, followed by IHC for 1 protein. In this example, probes for immune checkpoint markers PD1, TIM3, and PD-L1 were combined with a CD45 antibody as a marker of immune cells. As can be seen here for core 1A3, PD-L1 mRNA is visible in green, TIM3 mRNA is shown in yellow, and PD-L1 mRNA is visualized in red. And so these are 3 RNA targets that are combined with 1 antibody, CD45, and CD45 is shown in white in this image. We can also look at a close-up of this selected area, and it looks like this. So you can see that there are cells co-expressing LAG3, TIM3, and CD45. So these are LAG3 positive, TIM3 positive immune cells. They're also pointed out by the red arrows. You can also see green arrows, and they are pointing out PD1 positive, TIM3 positive immune cells. And there's also white arrows in this image that point out PD1 positive, LAG3 positive, and TIM3 positive immune cells. So these immune cells are positive for these 3 checkpoint markers. In this final section, we will switch gears a bit and discuss immune infiltration and state of lymphoid and myeloid cells in the tumor microenvironment. In order to assess the infiltration of selected lymphoid and myeloid cell populations in non-small cell lung cancer, the expression of several key functional markers such as FOXP3, interferon gamma, CXCL10, and CCL22 were evaluated in addition to key cell lineage markers like CD4, CD8, CD68, and CD163, and this resulted in the following probe sets. So we have 2 sets for lymphoid cell types beating CD8 plus cytokine interferon gamma, with double positive for these 2 targets being indicative for the presence of potential significant cytotoxic T lymphocytes. Also, we have a combination of CD4 and the transcription factor FOXP3, where double positives could be indicative of regulatory T cells. In addition to that, we have 2 sets for myeloid cell types being CD68 plus the chemokine CXCL10. Both markers are known to be expressed in several cell types, including M1 macrophages. Then the second combination that we have for myeloid cell types is CD163 and the chemokine CCL22, which are markers preferentially expressed in alternatively activated M2 macrophages. In this highly inflamed core 2E2 that is shown in this panel, we observed abundant infiltration of both lymphoid and myeloid cells, and the recruitment of CXCL10 expressing immune cells in the local tumor region is clearly visualized. The presence and abundance of regulatory T cells, so FOXP3 positive, CD4 positive cells, can also be evaluated in relation to CCL22 expression in immunosuppressive local tumor environment. Again, in this slide, also the arrows point to the double positive cells for these probe sets. Then we also have this 2C4 core for non-small cell lung cancer, and that has low infiltration of lymphoid and myeloid immune cells, as you can see here, especially in comparison with this highly infiltrated core 2E2. Here you can see the quantification of infiltrating lymphoid and myeloid immune cell types in the TME of selected ovarian cancer tissues using the probe sets that we just highlighted for non-small cell lung cancer, and we ran these probe sets on 4 serial sections. The quantification is based on the number of cells with positive dot signals for these targets. In this and the upcoming slides, we will compare the highly inflamed core C2 that we can see in the table over here with the low immune infiltrated core A1. Representative images for core C2 illustrate the high lymphoid immune cell infiltration with interferon gamma positive, CD8 positive cytotoxic T lymphocytes and CD4 positive, FOXP3 positive T regulatory cells. So these are the 4 panels that you can see to the left with a higher magnification image for each of these 2 probe sets that visualize lymphoid cell types. Then we have also CXCL10 expressing CD68 positive M1-like macrophages that are also abundant in this tissue core, which you can see in the 3rd column with again a magnification of the selected area for double positives that you can see for CXCL10 and CD68. Then there's also medium infiltration of CCL22 positive CD163 double positives, as they are indicators of M2-like myeloid cells, and that's what you can see in the last 2 panels. We will look in more detail at this highly immune infiltrated ovarian core, and that's what you can see over here. So here is a substantial area of core C2 with CD8 positive interferon gamma positive cells that are significant or that could indicate cytotoxic T lymphocytes. A higher magnification for these detected lymphoid immune cells is also shown in the left bottom corner, what you can see over here. Shown here is the same area of a serial tissue section with CD4 positive, FOXP3 positive cells, and these double positives are indicative of T regulatory cells. A higher magnification also for these detected lymphoid immune cells is shown here in the left bottom corner. The 3rd serial section was probed for CD68 in green and chemokine CXCL10 in red, with double positives that could be indicative of M1-like macrophages. If we look at this higher magnification for the selected area over here, you can see that there is a high degree of myeloid immune cells shown as the double positives. This 4th serial section is probed for cell lineage marker CD163 in green and chemokine CCL2 in red, with double positives indicative of alternatively activated M2 macrophages. Again, a higher magnification is shown for this selected area, and you can see the double positives for CCL22 and CD163 over here. In this slide, a summary of the highly inflamed core C2 can be seen on the top row, and you can compare it this way with the low immune infiltrated core A1, which is on the bottom. And this is shown for each of the 4 probe sets, representing the 2 lymphoid and the 2 myeloid immune cell types. And you can clearly see the difference between the high infiltrated core C2 on the top, both for lymphoid and myeloid cell types, when you compare it to the low inflamed core A1 on the bottom. And so they clearly visualize the numerical values that are seen in the table. Co-expression patterns indicative of lymphoid and myeloid immune cell types in non-small cell lung cancer were also visualized by our multiplex fluorescent ISH for 3 targets, and that in combination with IHC for 1 protein. So in this example that I will show you, we used probes for CD4 mRNA, FOXP3 mRNA, and interferon gamma mRNA, and it was combined with the CD8 antibody. So this is an example image for a non-small cell lung cancer core. And you can see the staining pattern with the probes marked as follows. So the CD4 probe or CD4 mRNA is shown in green, FOXP3 mRNA is shown in yellow, and the interferon gamma RNA is shown in red. and the signal for the CD8 antibody is shown in white. And so if we look at a close-up of this specific area in the center of this core, and also for the other selected area to the bottom, we can see it as follows. So the overlay for all these 4 channels is always shown on the left, and then we have the single channel images next to that. So what you can see here is we have red arrows pointing to these CD8 positive interferon gamma positive cells, so presumably the cytotoxic T lymphocytes. Then we also have an example where you can see CD4 positive and FOXP3 positive cells. So these double positives are indicated by the green arrows, both in the overlay and in the single channel images. And so these are double positives for CD4 and FOXP3, indicative of these T regulatory cell types. Thank you for attending this webinar, and I will be happy to take questions from the audience now.
Excellent presentation, Dr. Laeremans. Thanks for bringing that information to us. Before we get started with all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. Our first question is, what is the recommended thickness for FFPE samples?
So for FFPE samples, we recommend to use a tissue section with a thickness between 4 and 6 micrometers.
Can I use dot size to analyze target expression?
So no, you cannot. So the size of the dot here depends on the number of double Z pairs that is bound to the target sequence, as we showed in the beginning of this slide deck. And the bigger the dot size, that means that the more Z pairs are bound. The standard probe design is 20 Z pairs binding to a 1 kb long target sequence. But if a minimum of 3 double Z pairs is bound, you can already see the signal. So the number of dots is indicative of the number of transcripts, so it's 1-to-1, but the dot size should not be used to quantify the number of transcripts.
Can expression be quantified in different compartments for comparison, for example, tumor versus stroma?
Yes, that can definitely be done. So data can specifically be analyzed in selected areas of interest. So a tumor and stromal area can definitely be selected. We have also done this type of analysis, for instance, for LEG3 and PD-L1 expression in tumor versus stroma for some specific cores. And the data has been shown also in previous webinars and can be seen through the website. And in this specific case, we could see, for instance, that LEG3 expression was higher in the stromal area for some cores, while PD-L1 expression was higher in the tumor area. So definitely stromal and tumor compartments can be analyzed separately and compared.
How many RNA targets can you detect with dual ISH IHC, and is there a specific order when combining these two assays?
So the number of RNA targets that can be used in combination with IHC is up to 4 targets, and it depends on which RNAscope kit you use, so you can use 3 or 4. It is also highly recommended that when you want to combine ISH IHC, that you first run the ISH assay, and then in the end you combine it with the IHC. So when you do it, you should work with a well-validated antibody that you already know that works in your hands. Also, you want to run the ISH assay with the RNAscope pretreatment reagents first, because this will ensure that your protein can survive the protease and other pretreatment steps that are necessary for the RNAscope assay. So if you still see signal when you combine the RNAscope pretreatment with the IHC antibody, then you can also proceed and do the dual ISH IHC. And also it's better to target high abundant proteins because of these protease steps. But again, when you can optimize the antibody with the RNAscope pretreatment and it works, then it should also work for the dual ISH IHC.
Can heterogeneous expression, as shown for PD-L1, be quantified?
Yes, sure. A heterogeneous expression pattern means that so the cells display a different range of expression, and that's what we've shown in this talk for PD-L1, for instance. So it can definitely be quantified. There's different ways of doing it. I will I can mention it briefly. So one of them is you can use a semi-quantitative histological scoring method. So we have ACD criteria that can be used, and these criteria are based on the number of dots per cell, and that way you can give a score to your samples. Also, image-based software can be used. So as you've seen throughout this webinar, we use Halo software, but other software can also be used to actually quantify the number of dots per cell or in a specific region of interest. So for this type of analysis, as mentioned, we use Halo. But a third methodology you could also use that elaborates on this software is that you can actually bin cells with different expression levels into different bins. So that way you can get a histogram with the percent of cells which in each expression category or bin. And that way you can very easily visualize the heterogeneous expression that you have in your tissue. So people who want to know more about how to analyze RNAscope data, there is a webinar that can also be accessed through the website that elaborates on these three methodologies and gives clear examples for singleplex, duplex, and multiplex staining scenarios.
Can co-expression be quantified?
Yes, sure. So again, if you want to look for co-expression of two targets, you can set the settings that you want for your two individual probes, and it will segment cells, detect the dots within the cell, and from that type of analysis, you can also get the number of cells that co-express these two targets. So it's definitely feasible.
Can my target expression appear as clusters, and will this interfere with my data analysis?
Yes, it could be. So you can imagine that if you have a very high expressor, so if you detect that mRNA and in combination with signal amplification, you could get a signal that appears as a cluster. So what you can do, at least in the Halo software, is you can set the parameters for what you define as a dot, so dot size and so on. And what it will do next is it will say, okay, you have this size of cluster, and it will break it down into the number of dots that fit with all the settings that you use to define a dot. So it's definitely feasible to still get a number of dots out of a cluster of expression.
Is the new V2 fluorescent assay different from your existing RNAscope V1 fluorescent multiplex assay?
So our V2, no, let me rephrase. So our V1 assay is primarily for fresh frozen tissue. It is also a shorter assay and contains all the necessary components to run the entire assay. Recently, we also launched the V2 version of the fluorescent assay, and this assay is TSA-based. And this assay is slightly longer, but it is optimized primarily for FFPE tissues. So V1 is mostly for fresh frozen, and if you want to move to FFPE tissues, you can definitely use the V2 assay. Or if you, for instance, have a low expressor or tissue that might have like high autofluorescence, we also recommend to use the V2 multiplex fluorescent assay.
Can dual ISH IHC be performed on an automated machine?
Yes, for sure. So we have the Leica Bond RX, and also now we have the Ventana Discovery platform. So these are two automated platforms that can be used to run the multiplex fluorescent assay.
Well, thank you. If we didn't get to your question today, we will follow up with you via email. I would like to once again thank Dr. Laeremans for her presentation. Would also like to thank our sponsor, Advanced Cell Diagnostics, for making it possible to bring this presentation to you. Today's webcast will be available for on-demand viewing through May 2018. You'll receive an email from us alerting you when it's available on demand and posted on labroots.com. You're welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. We will see you next time.